Bacteria are so powerful that they can live in the harshest climates of Mars. Did you know about it? If not, then worry not. We are here to tell you all about it. New research suggests that signs of ancient Martian life could be out there, or rather, hidden just beneath the Martian surface, safe from harmful radiation. Without further ado, let's get back to the video. As Elton John once sang, Mars ain't the kind of place to raise your kids. In fact, it's cold as hell. But new research suggests that Martian chill could allow bacteria to survive for up to 280 million years below the planet's surface. The finding raises hopes that traces of ancient life, or even viable organisms in suspended animation, could be found on the red planet someday. In the study, scientists found that an Earth bacterium, Deinococcus radiogerans, is so resistant to radiation that it can handle the equivalent of 280 million years of the radiation present 33 feet, 10 meters, below the Martian surface. The plucky little microorganism, which has been found thriving in nuclear reactors on Earth, could even last 1.5 million years on the Martian surface, which is constantly bombarded with cosmic and solar radiation. The key to this survival is Mars' dry, cold environment. When desiccated and frozen to -110.2 degrees Fahrenheit, -79 degrees Celsius, the temperature of dry ice and the higher latitude regions of Mars, radiogerans, become phenomenally, astronomically radiation-resistant, said study senior author Michael Daly, a geneticist and radiation biology expert at Uniformed Services University in Maryland. Resisting radiation. Radiogerans has long been known to be a champ at resisting radiation. It's found in the human gut and in many other places on Earth, and has even survived for years in the vacuum of space. The new research, however, is the first attempt to test the bacterium's upper limit of radiation resistance when it's in a desiccated state. Previously, scientists had found that the bacterium can withstand 25,000 grays of radiation when in a liquid culture, Daly told Live Science. For comparison, a dose of 5 gray would kill a human. Daly and his colleagues dried and froze radiogerans and then bombarded the bacteria with both gamma radiation and proton radiation, mimicking cosmic radiation from deep space and solar radiation from the sun. They found that dried and frozen radiogerans could survive a mind-boggling 140,000 gray of radiation. That's equivalent to the dose from 1.5 million years on the Martian surface and 280 million years, 33 feet beneath the surface, where the only radiation is from the radioactive decay within rocks and minerals. The organisms survive irradiation in 2 ways. Study co-author Brian Hoffman, a chemist at Northwestern University, told Live Science. First, they have multiple copies of their genomes, providing a backup for any bits damaged by radiation. Second, they accumulate large amounts of manganese antioxidants, which capture damaging molecules created by radiation. The capture of these molecules prevents damage to the proteins that do DNA repair for the organism. The DNA is organized to be repaired, and these manganese antioxidants protect the proteins that do the repair, Hoffman said. Life on Mars. Radiogerans evolved on Earth, where the atmosphere protects the planet and its organisms from the worst radiation. The bacterium probably evolved to withstand damage during dry periods, and the radiation resistance is just a side effect of that evolution, Daly said. Any Martian bacteria would have had to evolve in an environment without that protection, and likely would have had to evolve similar radiation-widespread liquid water for 2 billion years, so even if ancient life did evolve there, 280 million years is still too short to suggest that the planet hosts a plethora of bacteria just waiting to spring back to life. But because Mars has a very thin atmosphere, meteorites rain onto the planet's surface regularly, Daly said. The heat and liquid water released by those impacts could potentially wake up dormant bacteria in the subsurface and allow for a temporary flourishing of life. Even if this temporary oasis theory isn't true, the long-lasting potential of bacteria on Mars means that bits and pieces of ancient life could still be present as traces in the rocks, Hoffman said. DNA and other signatures of life could exist as fragments, even if the organisms were long dead. The findings also have implications for preventing Mars from becoming contaminated with Earth bacteria, according to the study authors. Any D-radiogerins that hitched a ride on a Mars rover would likely survive the trip from Earth to the red planet. Other microorganisms, such as Escherichia coli and some Bacillus species, could also last thousands of years on the Martian surface if dried and desiccated, the researchers found. As more missions aim to return samples from Mars to Earth, it will be important to ensure that Earth microorganisms aren't accidentally hauled to Mars and then mistaken for extraterrestrials, said John Rummel, a senior scientist at the SETI Institute and former planetary protection officer for NASA. Rummel was not involved in the new study, but he oversaw its editing at the journal Astrobiology. The round-trip tourist organism is a problem here, Rummel said, and we have to be careful about how we deal with that. Microorganisms may be able to survive just under the surface of Mars for far longer than researchers thought. In a new set of experiments, researchers found that dried-up bacteria could stay alive in Mars-like conditions for up to 280 million years, which would improve our chances of finding evidence for them if Mars ever hosted life. Most previous experiments to test whether microorganisms could survive the intense radiation on Mars have used hydrated bacteria at room temperature. The longest-lived of those were thought to be able to survive for up to about 1 million years. Brian Hoffman at Northwestern University in Illinois and his colleagues took a different tack, desiccating a variety of bacteria and yeast and freezing them before subjecting them to radiation similar to what a buried life form on Mars would experience and measuring the damage. If the freeze-dried bacteria were warmed up and exposed to water during that time, they would emerge from their dormant state. It opens some possibilities that you could get revitalization if a meteor with some water comes down and splashes on the surface. It could regenerate, says Hoffman. The probability that it's still alive now has increased from 0 to the tiniest thing you could imagine. It's non-zero, but it sure as hell ain't big. It is estimated that Mars dried up about 3 billion years ago, so even the hardiest of bacteria are most likely dead by now. This longevity could make it easier for us to find preserved evidence of any bacteria that once lived on Mars, but it also means that if we contaminate it with earthly organisms, that contamination would be practically permanent. If we contaminate the area that we land in, how do we know whether what we find was there before we got there or whether it was something we deposited? Says Hoffman. If we can't tell what's ours and what's theirs, so to speak, then it's impossible to tell whether there was life there. That's it for the video. If you enjoyed it, kindly like the video and subscribe to our channel.